Assalamualaikum warahmatullahi wabarakatuh. In this occasion, we are going to present about our group discussions of Setora nitens in oil palm pest monitoring programs. And here are our group members.
Hello, my name is Mohamad Hafidh Adityawan with student number 202.
My name is Eksita Juliane with student number 204.
My name is Alifiandra Putra and my student number is 208.
My name is Muhammad Akbar with student number 210.
My name is Humam Abdussalam, 215.
My name is Dian Pamungkas with student number 217.
Okay, so for the first slide is we are talking about pathogen identity. The pathogen itself is called Setora nitens, and the host is oil palm or Elaeis guineensis chalk. Cause defoliation resulting in decreased FFB production fresh fruit bunch. Description of the pest. This is a serious pest of oil palm, as the caterpillars can strip the canopy bare due to their voracious feeding habits. Eggs of this pest are laid in mass on the underside of leaflets. Incubation period for the eggs is 1 to 18 days. The eggs then hatch and the larval period will last for about 42 to 55 days. A fully grown larva at the later stage can measure up to 35 millimeters. Larvae then enter the pupal period that lasts for about 17 to 31 days. The spherical brown cocoons of this pest are usually found at the base of the host plant. This pest is also generally kept in check most of the time by natural enemies.
Pest monitoring is the cornerstone of a pest management program. It's the first defense for successful pest control in an agricultural system. The idea of pest monitoring is to monitor the pest population over time so we can detect and predict the onset of a pest outbreak and take action before the problem worsens. Next. Thus, we can implement not only a more site and pest-specific treatment, but also with proper timing to the most vulnerable stage of the pest life cycle. This will help improve the success of pest control and at the same time reduce the amount of chemicals used to control the pest, which is the main concern of the integrated pest management concept.
Okay, one of the monitoring methods is by conducting a census of pest population and or damage by the pest. There are basically three objectives of a census program, which will determine the intensity and design of the census itself. They are detecting presence of a target species to prevent the spread of the species and pests with zero tolerance or very low threshold, such as rhinoceros beetle on young plantings. This type of census is an extensive program and covers a widespread area. The second one is providing information on the status of a target pest. This type of census basically is to know whether the population is below or above the threshold. Although the mean density of the pest is not really necessary, detailed information on the estimate of population intensity and stage of the pest is important to help us make a decision whether or not and when to do the treatment. Leaf-eating caterpillar and rats fall into this category. And the third is providing accurate density estimate with high degrees of precision. So change in population density of pests can be measured. This type of census needs intensive work and can also be implemented to measure spread of pests and disease that attack individual palms, such as termites and ganoderma. In this case, the exact number of infest or diseased palm is needed. Okay, we move to the loss incurred. As for the loss caused by the caterpillar pests are disrupting plant photosynthesis, because the leaves become dry, the midribs hang and ultimately have an impact on not forming bunch for 2 until 3 years. These pest larvae damage plant by eating oil palm leaves, generally starting from the lower leaves to the young leaves. This pest attack can result in defoliation, which results in a decrease in the production of FFB, fresh fruit bunch, by 40 till 60%.
Okay, so based on our group discussions, we decided to make this monitoring plan. The place will take place in Meadow Wartani Garden, Ngemplak, Sleman, Yogyakarta. The objective is to observe the intensity of Saturanitan pest in palm tree, and the times for this monitoring will held on 1st January until 1 April 2021. The interval will be done once every 2 weeks. Sample size will be 4 plot in 5 hectare with 136 trees for each hectare with 20 hectare with square sampling methods. The monitoring will be done in preliminary and the main survey. The preliminary survey was carried out in the form of an inspections of the research locations as well as interviews with farmers. Interview aimed to find out informations about the conditions of oil palm cultivations. This data was collected using a questionnaire at each sample locations, then determine the area of for oil palm plantations that met the criteria as allocations for observations and sample plan to be observed.
Main survey activity carried out. In the main survey, praise are first, photographing matter related on caterpillar pests, such as caterpillar droppings, caterpillar coconut, caterpillar larvae, and leaf symptoms caused by caterpillars. Second, determine the location of the sample points to be observed based on the land agreed by the farmer. Next, land sketch. The observation plots for each location were determined by survey method in one block of the field, namely Block A, with a total garden area of 20 hectare of yielding crops. The location of the observation was taken randomly as such as 5 hectare, so that are 4 plot location. On each hectare, there are 136 oil palm with plants with a 5 cropping patterns, X meter and 9 meter.
Determination of the sampling point or plant samples was carried out using the square method. In each sample plant, identification of the types of caterpillar pests that attack, the number or population of pests, the part of the plant affected, and the level of damage due to the attack is carried out. Meanwhile, the percentage of caterpillar attack parameters can only be seen whether there is an attack or visually on each oil palm plants in each location. Next. Observation of attack intensity was carried out visually based on the symptom of caterpillar pest. Each plot was taken 100 points to be observed, one plot of 5 sample point and one sample point of 20 point. Count the main plants that have visible symptom of attack one by one, then count how many of the 100 plants are affected. Next.
Okay, that's it from our groups. Thank you so much for watching. I hope our discussion may be helpful for you all. Wassalamualaikum warahmatullahi wabarakatuh.